Good morning. In my last session, we were discussed up to the determination of dosage for proving. Today, let us discuss about the proving team. The proving team consists of master prover, otherwise called project director, advisor, otherwise called expert, proving supervisors, otherwise called panel of investigators, and provers. Now, let us see about master prover, otherwise called project director. The master prover is the chief person responsible for the entire proving project. The master prover should be a qualified homeopath and familiar with homeopathic principles and methodology of proving. He should have sufficient practical experience in the process of proving. The master prover should have actively participated in the completion of proving. He should have enough experience in the supervision of proving and also participate in the post-proving compilation, analysis, collation, extraction and publications of provings. He should plan the entire proving, right from the pre-proving protocol to the post-proving task of analyzing the proving. He should ensure that the methods followed to conform to the highest standards. The master prover decides on the protocol to be followed and decides upon the drug and its potency. He coordinates with the different groups of people involved in the proving. He is responsible for the safety of provers, the quality of work of supervisors, the extraction, collation and editing of the symptoms. The master prover should be closely familiar with all the details as well as have an overview of the entire project. To ensure the freedom from prejudice, the master prover should be blinded to the remedy that is being proved and to the provers. Now, come to the advisor. Advisor is called expert. The advisor assists the master prover and provides him information regarding the details of the drug to be proved, its toxicity, both in toxic and non-toxic doses. The advisor may be a qualified botanist, zoologist, chemist or microbiologist, providing the characteristics of the drug to be proved. He also provides information about pharmacological action of the drug as well as the biological study of the drug on laboratory animals. The advisor may be a qualified physician who certifies the health of the prover and conduct necessary examination and investigation into the prover's health before, during or after the proving. Now, coming to the proving supervisors or panel of investigators. The proving supervisors or the investigators are the crucial and vital link between the prover and the project director. Supervisors also should be a qualified and experienced homeopaths. The proving supervisors monitor the responses and the records of the prover, inquiring in details into each symptoms recorded in the prover's daybook. Good attentive supervision is one of the key factors in ensuring successful and fruitful proving. The proving supervisors preferably should have the experience of participating as provers in previous provings. They take the prover's case before the proving and need to keep a close contact with the prover from the moment of the first dose and have to be available for the eliciting symptoms thereafter. They need to pay the utmost attention and receptive to any change in the state of the prover and make sure that it is recorded in the prover's notebook as well as in their elaboration book. Supervisors have to notify the coordinators of any changes in the state of the prover that might potentially affect the safety of the provers. The supervisors needs to maintain the same fidelity in eliciting the right, true and complete picture of the deviations as noticed by the prover as is required in tracing the portrait of the disease. The demands freedom from prejudice, sound sense and attention in observing from the proving supervisors. They also need to be blind from the medicines that is proved and also from the provers that receive the test dose to obtain a pure, unbiased proving record. Now come to the provers. The best possible way of ascertaining the pure, peculiar effects of drug is by producing drug on healthy human beings. These healthy subjects on whom the drug is tested to obtain the portrait of the medicine are called provers. Now let us see about the methodology. Proving requires a thorough planning and study before and after the proving. Hence, the proving experiment is best studied under 3 different protocols. The first protocol is the pre-proving protocol. The second one is the proving protocol. And the last one is post-proving protocol. Now come to the pre-proving protocol. Here, first we look into the study of proposed test drug. The proving master undertake the study of the drug covering areas of the pharmacognosy, standardization, pharmacology, and toxicology. He should also find out where there is an antidote available to the test drug. The proving master decides on the test drug and he is responsible for its procurement from a reliable source. The proving master undertakes the project of proving either a new drug or a reproving of existing drugs from the materia medica. Now let us see about the selection of supervisors. The proving master selected the panel of investigators or supervisors and instructed them about the ethics and conduct of proving. Supervisors also should be qualified and experienced homeopaths, and preferably he should have the experience of participating as provers in the previous provings. The supervisors prepare the initial medical report pro forma. In the case of selection of provers, only healthy individuals are selected as provers from different regions of the country to ascertain whether ecological, socio-economic, climatic, regional factors and food habits. An undertaking is used to take the consent of the provers. The consent form having the name, age, sex and address of the prover. Persons selected as provers must be between the age group of 18 to 45 years. The provers are intelligent enough to understand the seriousness of the experiment and he can record the subjective symptoms properly. The drug must be proved on both male and female to get changes produced in the sexual sphere. Now come to the ethical considerations. Consent should be obtained in a writing form from the subject, but the responsibility always remains with the investigator or investigating team. The responsibility never falls on the prover even after the consent has been obtained. The nature and the purpose of drug proving must be explained to the prover. The investigator or investigating team should discontinue the provings if it would be harmful to the prover. Proving should never be done in toxic doses. Actually, for toxic symptoms, we must exactly depend on the reports of accidental provings recorded in the toxicological literatures. Now, let us see what are the criteria of a prover. The person should be responsible, reasonable, healthy, and well balanced in body, soul, and spirit. The prover must be able to lead a life that is normal as possible during the proving. Keep a definite pattern in his daily life with a fixed time for sleep, work, eat, etc. The prover must be intelligent enough to properly record the subjective symptoms as deviations from his normal conditions of life. These subjective symptoms are the highest value. Honesty is a qualification of a good prover, for he must be very careful to record all phenomena from the very beginning of the trial. Persons who are hysterical or displaying anxieties and emotionally disturbed stated should not be included in the proving trials. Persons suffering from allergic manifestations, particularly on the respiratory system and skin, also should not be included. Those who have a lot of emotional symptoms should not be included in the proving trials, because too many emotional symptoms may be confuse the final results. Now, let us see what are the instructions are given to the prover before starting a proving. Never make any major changes in their daily routine and lifestyle, specifically food habits, sleep, exercise, etc. during the proving period. To mention in the prover's report about any probable precipitating factors which could have been produced the symptoms, such as overexertion, night watching, any excitement, excess intake of coffee, tea, overeating, indiscretions in diet, etc. Now, come to the primary coding of a remedy. The proving master is responsible for the primary coding of the remedy. The test drug as well as the control placebo should be coded. The doses that are received by the prover also should be numbered. The provers also assigned code numbers. The coding should be done by committee, and that shall not be involved in the direct supervisions of the records of provers. A detailed record of the coding should be maintained, that is unlocked after the initial blind assessment at the termination of the actual testing of the drug. Now come to the proving protocol. Hahnemann recommended the preparations of the test drug by the actual provers so that they can trust on them. The provers were hence fully aware of the nature of the substances that they were proving. But in the case of modern proving methodology, of this concept is totally changed. The entire modern proving process are more specifics. Now let us see what is the multicentric trials. The multicentric trials are undertaken for the proving of a drug, keeping all variations of the prover's lifestyle, food habits, constitutional and temperamental factors, and climate. The studies should be conducted at least at 3 different centers under the same protocol before releasing the data for the professional use. Ideally, the proving should be conducted at 3 different locations, such as in the mountains, on the low plains, and at the seashore. Now come to the orientation meeting. Orientation meetings are arranged at the commencement of the proving. In this meeting, explain the importance and the conducts of the proving process in details to the provers and supervisors. Instruction letter should be given to the prover and the supervisor. They should study it carefully and familiarize themselves with details of the instruction letter. Any queries regarding the nature, process, outcome, complications, etc. should be cleared at that meeting. In this meeting, the day book are also provided to the provers for the recording of the proving. Now let's come into the nature of trials. Since 1948, randomized controlled trial, otherwise called RCT, has become the standard designs for drawing valid conclusions on the efficacy of the medicine. In the case of randomized trials, those in which the subject of an experiment are randomly placed either in treatment groups or in placebo group. The researchers attempt to place people with similar characteristics in equal numbers in treatment and placebo groups. This process helps reduce the bias. In case of double-blind trials in which neither the experimenter nor the subject know whether a specific treatment was prescribed or a placebo. Placebo means a fake medicine that looks and tastes like a real homeopathic medicine. The double-blind modifications in proving trials wherein neither the proving master nor the prover know the name of the drug and its potencies of the drug or the control received by each the provers. In India, the authority has adapted Hahnemann's concepts of proving of drug on healthy human volunteers with modifications based on double-blind techniques. The crossover design is followed during the entire course of the proving. Test drug as well as the control is coded. The different doses that are received by a single prover are also numbered. The provers are also assigned code numbers. The coding should be done by the committee that shall not be involved in the direct supervisions of the records of the provers. Blinding also signifies the blinding assignments of the codes. A detailed record of the coding should be maintained, that is unlocked after the initial blind assessment at the termination of the actual testing of the drug. In case of crossover studies, otherwise called double control studies, experiments in which half of the subject of a study are given placebo during one phase of study and then given an active treatment during the second phase, while the other half began with the active treatment and then received placebo during the second phase. Crossover studies sometimes do not test a placebo and instead of compare one type of treatment with another type of treatment. In case of controlled trial placebo, the use of placebo is aimed at giving a picture of the remedy response, separate from the physiological, psychological and emotional aspects of the doctor-patient relationship. A trial can be described as controlled only if it included a method of eliminations. The controlled medicine must be a similar preparation that is identical in all other respects except there is no other original and specific substances to be mixed and potentized with the vehicles. It would be ideal to titrate or success the placebo doses to remove bias following the potentization of the vehicle. That's all for today. I will come again tomorrow with the remaining portions. Until then, goodbye. Thank you.